Hi there, and welcome to a video showing the major steps of phagocytosis. So I've shown a white blood cell here. I'm using a macrophage. Again, there's various subcategories of macrophages and other phagocytic cells. The first step is for the white blood cell to move to the location of the foreign material. I'm going to use microbe here as an example. So there's different chemicals that can alert it to the fact there's a microbe there. A set of important chemicals are called PAMPs. And PAMPs are short for pathogen-associated molecular patterns. So that's kind of a mouthful. So pathogen sounds fancy, but kind of makes sense when you look at some examples. So these are materials that are foreign to the body. So there's something strange or unusual. It's not something that is found in human cells. So one of those is the LPS, or lipopolysaccharide layer, that's found in the outer part of the envelope in gram-negative cells. Peptidoglycan, which makes up layers of the cell wall in gram-positive and gram-negative cells. Also kind of unusual, something that we have but not in this form, an unusual form, something like double-stranded RNA that's seen in viruses. So just some examples there of some things that kind of alert the white blood cell system that this is not part of us, this is not one of us. Interestingly, in what we eat, you would think that would, we're taking in all these different substances. The body does seem to recognize that normal food substances that we consume are not harmful, although we know that can take a different path when people have food sensitivities or allergies. So those are some of the things that would kind of alert the microbe as well. And there's different chemicals that are released during inflammation and other things to enhance chemotaxis. So going back to our cell surface receptors, they are actually going to, and again, this is not all to scale and not to detail of how it's quite happening in terms of the binding, but if this were my microbe here, it's going to bind to the microbe, and this stage is called adhesion, and I'm going to lump it together with the next stage and ingestion. So it's binding, so adhesion is pretty self-explanatory, as well as ingestion. So steps 2 and 3. And it brings it in what's called a phagosome. And so this membrane, get my colors here, is going to kind of fold in and pinch and bring in this microbe. There's our microbe again. Okay, so it'll bring that in, and this is called phagosome formation. And we're just going to write phagosome here for short. So now it's got it, it's brought it in. So now we want to destroy it, now that it's in. So this phagosome is going to fuse with another vesicle that many of you are familiar with called the lysosome. And the lysosome carries various, lysosome. It carries various degradative enzymes and compounds. And so as that fuses with it, this is now called, there's our microbe again, a phagolysosome. Okay, and so then the idea is that there is destruction in there. And so some of the things that might be in there are oxidizing compounds, nitric oxide, as well as various enzymes that are degradative, proteases, nucleases. And so that is going to then ultimately, look for my colors here, go through this one of the later steps, which is destruction. So it's hopefully going to take this microbe and just break it up. And then finally release on the outside. It's going to dump these microbits out there. Now some of them can be retained for what we call antigen presenting, which I'm not going to diagram here. but the other parts will be released. So some microbes can survive this process, so maybe they have a capsule or they release kind of counteractive chemicals that prevent it from being destroyed. So sometimes if a microbe has, let's say, a capsule coating, they go through these steps, but the enzymes they're exposed to aren't enough to actually kill the living cell. So there's different ways that microbes can kind of get around this. Some microbes release chemicals that actually damage, they're called leukocidins, they actually damage the white blood cells. So there are some microbes that can get around this, but again, this is part of that second line defense, so it's a generic response. It's not a response to, one type of microbe, it's any type of microbe, and it takes it in this way to do that. Okay, I think I have everything. Oh, a couple other things I wanted to mention. There's some other kind of related compounds that are mentioned here as well. Toll-like receptors that also can be found on the surface of white blood cells, and those are simply immune system stimulators. I'm not going to go into the details of that structure or process, but I mentioned in other videos that there's a lot of cross stimulation because you're really trying to ramp this system up. There's another group of compounds called collectins that also bind to microbes as well, but they are not attached to a cell, but they help stimulate. I mentioned the chemotaxis and that adhesion, so it helps stimulate phagocytosis. And these are just free-floating chemicals within the white blood cell system. Okay, so that's kind of an overview. Great animations on this process that really show all the steps and the breakdown, and a little bit more reading you can do on some of the ways that microbes evade phagocytosis or come through it unscathed, if you will. That's all I have on this. Please contact me with any questions you have. Again, this is Corey Andres, Central New Mexico Community College, using CNM Glass.